Good morning, students. This is for the fifth semester student, continuing the chapter of how extraction, isolation, and characterization, in which we are coming to start with the second session with the chromatographic techniques and various instrumental techniques also. We cannot go in much detail, but at least they have given in a full chapter to be covered with all the sessions actually. So let me go for this isolation and characterization technique. Extraction we have already discussed in the last class. This we will just part by part, one sessions we will have a discussion about this for the fifth semester, pharmacognosy and phytochemistry 2 of the B Pharm students. This may be useful. Isolation and characteristic techniques which we are going to discuss, there are many, but which we are going to discuss according to the syllabus and the requirements for the contents which we can cover is chromatography. In which column chromatography we are going to have a discussion, how the packing and what do you mean by small applications and all. We are going to see in herbs how you are going to isolate the compounds. And paper chromatography, thin layer chromatography, then high performance liquid chromatography, and gas liquid chromatography, and then high performance thin layer chromatography, HPTLC, which is having more application in pharmaceutical industries, and spectra study. There are many other chromatographies, but we are going to concentrate in this. Even I have added some more techniques also, like spectroscopic studies. We have to see about the UV spectroscopy, not going in much detail, but still we should know about. You will be studying in detail about instrument and analysis chapter subject or in a semester. So anyhow we should know about the instrument, what is its application in the in our pharmacognosy and herbal drug technology, and infrared spectroscopy, what's its application and how the instrument, just in principal part, and as well as we should know the applications, mass spectroscopy and nuclear magnetic spectroscopy, NMR, and also we have to see about electrophoresis techniques. Also we are now advanced. Apart from that, there are many instruments are there, but still we are just reducing with this one. And also how the hyphenated, that means two instruments to join together also, and two-dimensional, three-dimensional, this discussions also slightly we are going to make in the various classes. And also we have to make about some discussion about the electrophoresis, like paper electrophoresis, gel electrophoresis, and capillary electrophoresis. And also we are going to see about the electrophoresis, how it is applicable for in the DNA fingerprinting area, how in various forensic departments, how they are performing about this DNA analysis, which is, you may be seeing about the common criminology, what they are doing. That is also we are having a slight discussion how it is applicable for the herbal drug technology and isolation of compounds from natural sources. And here we are going to see what is chromatography. Already we know that is in other subjects also you might have studied. I am just going in a small explanation, and it may be helpful for you in other subjects also, which you can incorporate whichever the knowledge which you have. I am giving you a small idea and small definitions on how to do the things. And you can see chromatography is a technique, if you see, widely used for the separation and identification of compound of mixture. Otherwise anyone can say, and definition whoever says, suppose what is chromatography means, chromatography is a technique which is used to separate mixture of compounds. And chromatography is also very suitable for the extraction of plant extracts That is extraction of plant extracts or isolation of plant extracts, especially when a small amount, very small amount, minute small amounts can be go for TLC, paper chromatography, and also HPTLC, high performance thin layer chromatographies are available, which can be very minute quantities can be separated, and even HPLC also, small amount of material is supplied. And when the component is closed, even very close compounds, for example like alkaloids, androquinone glycosides or androquinones, or which are having chemical structures which are having something phenol or double bonds, whichever having something ring structures which is similar to certain compounds, can also be easily separated and identified. That is what they are resembled compound like alkaloids and androquinones are the examples which they have been separated by these chromatographic techniques. And in the earliest form, chromatography was applied only to colored compounds, because visible compound, colored compound which can be identified. And then the development of the chromatographic techniques increased even the colorless compounds by means of detecting liquids, and also by means of using various techniques. Even instrument analysis can also be done, and also by means of using UV spectroscopy, fluorimetry, according to the compounds whichever having the reactivity with the light radiations can also be done, and by connecting. So that's what colorless compounds. In the earlier stage form, the chromatography was applied only for colored compounds. That is what we discussed. But now, and the resulting of the separation is visual observations have been done. But now there are many. Understand? That's what I want to say. But for example, when you see the chromatography, this is also not very good classification. But anyhow, for study purpose, I have made this one. Even you are unable to see the slide properly, that's why I'm just pulling down little so that you can see the classification properly. So see here, it shows that the classification of chromatography can be done by in the three ways. There are many. Simply for on the basis of interaction of the solute and the stationary phase. There will be a mobile phase and the stationary phase in every chromatography. So by that you can see adsorption chromatography, partition chromatography, ion exchange chromatography, size exclusion chromatography, that can be called as even gel permeable chromatography. So there are many chromatographies on the basis of the stationary phase, and affinity chromatography, and you can see even proteins when you are using ligands, how the affinity or the attachments of the ligands, and separating the compounds by means of what is the stationary phase and the mobile phase, what is its basic reactions by means of the principal part of the chromatographies can also be differentiated. Specifically, mainly we can differentiate commonly in everywhere they will be saying adsorption and partition chromatography. Apart from that, there are many, what is the basic reactions between, or basic separation techniques between the stationary phase and the mobile phase plays the major role to separate many types of chromatographic techniques. And also on the basis of chromatographic bed shape, and you can see how the bed shape. So for example, what is the shape of that? For example, it is maybe one dimensional, two dimensional, three dimensional. Directions of the movements of the stationary phase can be changed in the axis. For example, x-axis or y-axis and z-axis can be moved, and also the separation can be more efficiently, more related compounds, more a sequence of very near structural compounds can also be very easily separated on the basis of the chromatographic bed shape, like two-dimensional, three-dimensional. It can be done in TLC, paper chromatography, and also even it can be done in three-dimensional chromatographies, can be done in the large scale of column chromatography also. It is having various techniques are to be followed. And on the basis of physical states, in the books they have made a mistake. Even I have shown in the last classes also I have shown, I think it's what they have shape. It's not shape, it is the second one is shape, and the third one is states. What is the states? That is, for example, liquid, solid and gas states are the many things which you don't know. So which we know that, or not don't know, we know that is there are liquid, solid, gas. These are the three states which is available for the matter. So we can see liquid chromatography, liquid chromatography, which is also working in the partition chromatography, but liquid and solid is also adsorption chromatography. So liquid chromatography, there are pure liquid chromatography and also gas chromatography usually can be separated. And also you can see there are gas solid chromatography, and also you can see solid and the liquid chromatographies are also there, which like you can see when the stationary phase is solid and the liquid is the mobile phase, then we'll say it's not working under adsorption chromatography. When the stationary phase is also liquid and the mobile phase is also liquid, then we will say it is working under the partition chromatography and liquid chromatography. So gas is also having the same principle behind, or same physical properties of the almost same like the liquid. So we can see gas liquid chromatography also work in the partition chromatography basis of separation. So by means of separation also we can classify, and the base of the shape of the operation also can be classified, and the state of the material which is used also can be classified. This is just to give you some idea about the chromatography which we had discussion in our class. So the students who are remembering about the definition of the chromatography, first, it is a technique which is used for separating mixture of compound. That's very important for you to know about it. And also now we are going to think about what is adsorption chromatography. Here I have given a picture just to show you. For example, you can see this picture shows a mixture of protein which is added to a column which contain, you can see there is a black marks, there are many types of shapes of colors are there. You can see this very clearly, even though it's not much clear for you because it's hidden. I'm just showing it. Just you can pull it down so that it is easy for the students to see. So you can see here. Now you see here, that is the proteins which you are, there is a mixture of proteins which you are interested to separate. So this is an example for adsorption or affinity chromatography which can be holded, and after some time adsorption is a surface phenomena, absorption is an internal phenomenon which is absorbed. This is adsorption. Adsorption means reversible. It can be removed by means of a solvent system. You can see the picture. There is a mixture of protein which is poured into the column, and we stated down there is another. For example, when you pour it as a mixture which you are interested to separate, what will happen? It will be just going inside, and the stationary phase, there are stationary phase which is kept as a gel. According to the size of the molecule, it is just going inside, and we are interested to separate one compound which is, the main thing is red color. You can see there is reddish, moon shape, square shape, and orange triangle shape are there. So red, you just imagine we are interested to separate the red. This is an example just to show for the student to understand. So when you are adding that, there is another black dots, there is another one solution which is ligand. So this ligand, you can see this ligand is having a capacity to hold that particular, that is a solvent system, or the ligand which is having a system which can hold or which can have a bind with the red color protein which is in the mixture. So you can see in the second side, when you are adding a solution in a separating funnel, there is a blue color solution as a mobile solvent, it takes all the other protein except that red color protein. Why? Because the red color protein have been made to stay there by means of using a ligand, or by means of using another compound which can hold it. So all the other things are removed in the first. You can see there is shown that 3, 4, 5, 6, 7 like that. There is some tubes which is shown down, that is called elute, the solvent which is removed. So first when you remove, you can see all the other, first itself, and the third which is shown itself, the blue color and the yellow color, triangle, square shape, they have already removed all the other proteins which are not holded by that particular ligand which is present in the stationary phase. But you can see in the 7th stage and also 6th stage, red color with a black spot which comes down. So there is afterwards, after they are removed, this alone comes because we are using another solution which can remove that ligand. And as a solution of the ligand, when you add the solution of the ligand alone will remove which is attached with it. So that you are changing the solvent system and you are changing the system, and so that you are removing alone, separating a mixture of compound. First initially it was mixture. It was the red color was along with the orange and also the blue color. But the orange and the red blue are separated initially because they were not holded. They are not having any affinity towards the ligand which you have added. So when one which is having attraction towards the ligand will be holded there, and one which is having more affinity will stay for a longer time, and then it can be separated. This is a technique of adsorption very easily. How the affinity towards the stationary phase of the molecules which is present in the mixture of compounds, if it is more, then they will stay for a longer time, and if it is not having any attachment with the ligand or with the stationary phase which you are using, it will be eluted initially. That means it will be taken out to the initially. So it is a simplest form of this method. The chromatography consists of passing a solution of a mixture of compounds needed to be separated, and you can see through a hollow glass column pack with a finely divided adsorbent powder and collecting the solution. The collecting solution is called elute. So that picture is just an example for you to show you. This is also another picture of column chromatography, which they have done colored separation. They have added a mixture in the top and they have passed through a mobile solvent, and you can see the picture with the color, difference in the color, light color, dark color, orange, yellow, and even a light cream colors are also can be seen, yellowish color. So that is the separation of colored compound in different bands, and these bands can also be removed from that particular column, and then you can use another solvent and then it can be separated or cut into pieces, or you can even use different types of solvents which can solubilize each and every one in one stage can also be used for separation of this compound. So the surface phenomenon of adsorption is utilized here, that is called adsorption chromatography. The finely divided solids are capable of selective adsorption to other substance. The components of a mixture introduced into a column of adsorbent are more or less strong adsorbed. So which is more strongly adsorbed? Those which are least strongly adsorbed will be carried down very faster the column by the passing the solvent, and the first one of the elute from the bottom of the column. So there will be one which is not at all having any affinity towards the stationary phase. They will be very fast will be eluted from the column at the bottom. Understand? That is what. So as adsorption chromatography, I have given an explanation. Again, the stationary phase is a solid, and the separation is due to the series of adsorption steps, and the passing of the more strongly adsorbed substances will go slower and less will be and they will be last eluted. The fraction of eluent, that means the substance which is coming in the solvent out and containing each component of the original mixture, can be then be separated and analyzed. So this alternatively the entire adsorbent core may be extruded, and after bands containing the compounds of the mixture have developed, as we seen in the color, each bands can also be cut into pieces, or it can be removed as a cylinder and also we can use, or solvents can also be used for their eluting. Individual bands can then be extracted by a suitable solvent to obtain each compound, and also we can use by means of TLC, or which can be used for identification of compounds in this column chromatographies and mixer with the two chromatographies. And the partition chromatography, the basic difference between, what do you mean by partition? It depends upon the solubility of any solute between two immiscible liquids. So stationary phase should be liquid and the mobile phase should be liquid. That you have to always remember. Adsorption means there should be a solid and there should be a liquid or a solid or a gas. And here you can see partition means there should be difference. Partition means the distribution or the solubility of a solute between two immiscible liquid is partition coefficient of a compound of a mixture which has separated between an aqueous and an immiscible organic solvent. It is very common. And for example, it's a lipophilic or a hydrophilic in nature compounds only, it will be immiscible. So immiscible liquids, so we can say organic and aqueous solutions which can be used. One can be organic solvent, can be as a stationary phase in the mobile phase, can be an aqueous, or aqueous may be a stationary phase in the organic, can be an mobile phase. The polarities can be easily changed, and also we can have various types of stationary phase and mobile phases can be used for our technique of separation using partition chromatography. The stationary phase is normally aqueous, which is mixed with an inert carrier powder and packed in a glass column, that is for column chromatography. So the mixture to be separated is dissolved in an organic solvent, is introduced into the column, and the chromatograph developed with more solvent or different solvent to increase the eluting power. And diffusion of the mobile phase through the stationary phase occurs. The different rates of travel of the constituents of the mixture are directly related to their partition coefficient between the mobile organic phase and the stationary aqueous phase. So the finished chromatogram exhibit a separated zone similar to those seen in adsorbent column, and this can be used by means of, this is the column which is, you can see, which is having a solid stationary phase with an liquid which can be stationary, solid supporting material with a liquid stationary phase. There also we can use a liquid as a mobile phase where the separation will take place by partition chromatography between the solubility. So you can see there just a picture to show you that is mixture of compound. When it is given, there is separation of yellow and red. So red, the solubility of the red towards the mobile phase is more, and it is, the distribution is very far, more. That is why the partition coefficient is more. That's why it came first, and the yellow is coming behind. So this is what, the depending upon the partition and the solubility of the solute between the two immiscible liquids, or between the stationary phase and the mobile phase, plays the major role for the separation of the compound is partition chromatography. And now we are talking about the techniques actually. Understand, that is about the principle we talked on. Now we are talking about column chromatography. Column. This is column TLC. These are all techniques. That is the principle. Adsorption and partition means there is a principle behind. The basic principle for any chromatography will depends upon these two. But apart from that, we are told molecular exclusion, and we are told about molecular exclusion and gel permeation chromatography and ion exchange chromatography. These all comes under, basically they are, and affinity chromatography. They all of them are adsorption, and partition is the basic principle behind it. And apart from that, there will be molecular exclusion, and these all will comes under it. And in a column chromatography, they used packed column or stationary phase as a stationary. The column may be packed with solid or a liquid. Liquid, as I told, if you want to hold the liquid in a tube, it needs a supporting material, and it can be a solid or a liquid. But if it is solid as a stationary phase and the mobile phase is a liquid, then it works in the adsorption chromatography. Liquid as a stationary phase and liquid as a mobile phase, then it will work as a partition chromatography as a basic principle. If the packing should be with a solid, and the principle of separation is adsorption. While the packing is done by the liquid, then the principle of separation will be partition. Very simple. The student can easily write about this principle behind, and also he can go for the working about various technique behind the chromatography. So generally in the column chromatography, the most separation is based on the adsorption principle. In adsorption chromatography, the extract which is to be analyzed should be dissolved in the mobile phase which you are using. Then it will be easy to, for example, in the column add. The individual constituents in the extract will move with different rates, and depending upon the affinity towards the stationary phase, if it is an adsorbent, understand, or the adsorbent, the compound which is more affinity towards the stationary phase will be separated in the last. It will take long time. It will stay for the stationary phase longer time because they have more affinity towards the stationary phase. While those which are less affinity towards the stationary phase, they will move faster and they will be separated first. This is the thing which we know very well, and it can be written very clearly as an important point. The stationary phase which is used to fill the column should be uniform, spherical shape or spherical size, and with high mechanical stability, inert with solute or other component, and mobile phase have free flow, free available, inexpensive, and available to separate the wide variety of the compound. Understand, that is what it's the main criteria which is required for a stationary phase when you are selecting in an adsorption column chromatography. Example, the weak adsorbent like sucrose, starch, inulin, talc, while medium adsorbent is calcium carbonate, magnesium oxide, calcium hydroxide, and magnesium carbonate. An example of strong adsorbent is activated alumina, activated charcoal, and fuller's earth, etc. So when you talk about activated, I hope the students can be known that activated means unwanted moisture which may be present in this absorbing materials like alumina and charcoal have to be removed. Then only it can act as a very good adsorbent for the separation of the compound in chromatography. That's why we used to use activate. Even the TLC plates, when you are made into for the action, we used to say activate. Activate means to remove the unwanted moisture or unwanted water which may be present, which will hinder our experiment for the separation of the compound. And the mobile phase may be used in the individual or can be used into the mixture depending upon their polarity and type of constituents to be separated. So you can just depending upon their polarity and the type of constituents. What type of constituents? What is the type of constituents? What is the chemical nature of the constituent you are interested to separate is needed for the researcher to go for this type of experiment to get separated in a column chromatography. The column should be made up of neutral gas or not glass, glass, and not affected by acid, alkali, mobile phase and the stationary phase. So this is very important criteria which you are using a glass. And the length of the column, length and the diameter ratio should be 10:1 or 30:1. Sometimes it may be 100:1. Long columns can also be used for a good separation or a very minute separations, or a large quantity of separations can also be, size can be varied. Small can be used and the big also can be used. So when you go for this packing materials for the packing of column. So you see this one, the picture is not to be seen very clearly. So just I am taking it to only 2 seconds to come down to make it clear. That is a funnel or a separating funnel can also be used in the place where the solvent should be added. I want to just to show you the picture clearly. That's why I just took this time. And you can see this picture, you can see you have a funnel in the first place, and the second one is, you can see, glass column, and the third one it is called eluent which you are collecting, that is eluent, that is a eluent which you are adding there. And then sand, you can see number 3 sand, and the 5 one is adsorbent, maybe silica or maybe alumina, activated alumina, and the glass will have to be kept in the 6th position before and also a filter paper can be kept even. And also in the top also we can keep for the sand and the filter paper for purifying the solvent system which you are using. Collecting flask, the 7th one is collecting flask. So you can see the 2 places we are keeping sand and the filter paper which is not shown we can keep. So packing of the column, the lower portion of the column should be packed with glass wool or cotton wool, to above which you can adsorbents can be packed as shown in the picture. And after packing with adsorbent, a paper disc, even a filter disc can also be kept in the top and then in the adsorbent layer, so that the introduction of the sample in the mobile phase will not disturb the adsorbent layer. So there is a 2 types of column packing. This wet packing, that means dry packing, and also wet packing techniques are there. So what is the difference between them and what is the defaults, which is very simple to discuss about that. And in the dry packing technique, the column is packed in the dry adsorbent, very dry adsorbent, which you are packed slowly in the column, and the solvent should be poured from the top to get the equilibrium, obtain the major drawback in this, air may be trapped up, because dry powder you are adding into the tubes, so air bubbles, air can be trapped and it can crack the column and between the solvent adsorbent and produces trouble in the separation of the compound. But in the wet technique, the mobile phase is mixed with the stationary solid powder adsorbent, and that can be poured into the solvent, so that there will not be any air can be removed when you are stirring by means of using any ultrasonic stirring or mechanical stirring or magnetic stirring or whatever maybe you can see, and the air entrapping can be reduced and comparatively less when it is compared to the dry packing, and uniform packing occurs in this column when you are using wet packing. If the flow of the mobile phase will uniform, there will be no development of crack, and a good uniform separation of the extract will occur. So the sample introduction, you can see the sample should be dissolved in at least a quantity of a mobile phase. You have to first dissolve, and completely sample should be placed in the top, or you can see top, you have to pour it in the column and it will be absorbed in the top area, and then the sample is isolated by eluting by using solvent system. And elution may be done by two methods. One is by means of isocratic elution, that means single solvent system which is using, but and the single polarity of solvent system. But gradient means increasing the polarities, for example by means of 10% to 80, 90%, and like that different solvents. For example, if you want to increase a petroleum ether, for example, if you should know the solvent system from the lower polarity to the higher polarity, as we discussed in the class, when it should be in the fingertip, like petroleum ether, benzene, chloroform, carbon tetrachloride, methyl acetate, or before that you can see acetone, and also you can see methanol, ethanol, methanol, and dilute acids. Like that you should know that how the increasing of polarity. Suppose if you want to increase slightly, you can gradient method means, for example, 10% of benzene and 90% of chloroform. When you add, automatically the chloroform polarity is slight for 90% of benzene and 10% of chloroform. The benzene polarity will be slightly increased, and then if it is 80% of benzene and 20% of chloroform, the benzene polarity will be slightly increased by chloroform. So like that, slowly gradient method also it is possible for isolating the compound, and different minute compounds can be differentiated and you can elute them very clearly by means of their affinity towards the stationary phase, if it is an absorption chromatography. If it is in partition chromatography, according to the solubility between the two immiscible liquids like stationary and mobile phase, the partition coefficient of the solute methods can also be easily used for separating very minute differences in the structural variation, can also be isolated or separated by means of this technique by liquid chromatographies. And also elution method, as I told, there is two techniques. One is isocratic elution, another is gradient elution. Don't forget, this will be coming even in the HPLC also. This is a very important sentence, how the separation of elution can be taken. So in isocratic elution, the composition of the mobile phase will be the same entire separation. There will be using only one solvent system which is used for the separation. That is called isocratic, single solvent system which is used for as a mobile phase to separation. But in the gradient elution, the combination of the mobile phase can be changed slowly, as I told, 10 to 90, 20 to 80, or 40 to 60, and 50 to 50, and 40 to 50. Like that, you can just increase the gradiently. Gradiently, you can increase the polarities of the solvent system, can also be used for the separation, changing the solvent system, slowly improving the separation. The polarity of this mobile phase can be changed by changing the mobile phase combinations, as I told, by means of increasing and decreasing the percentage of adding and increase the polarity. So detection compounds, the colored components can also be detected visually, and the colored bands moving in the column can also be collected separately. The colorless compounds can be detected by UV technique, refractive index detector, monitoring by TLC, by flame ionizing detector, or such other techniques. The component can be recovered by different techniques, sometimes even by cutting the columns, and the column may be made up of plastics and it can be easily cut into different zones or different distinct zones. But the best method is eluting, in which the component eluted by the mobile phase and the constituents is separated from the mobile phase. That is the best method which can be done. And now we will just have a discussion in the next class about the thin layer chromatography, and this will be continued for the next class. So we have thin layer chromatography, paper chromatography, HPLC, HPTLC, and also many classes are there. It will be compiled, and it should be studied very clearly for the separation of the compound. Thank you.